Meiosis is reducing the chromosome number for sexual reproduction to occur. So it occurs in the life cycle of sexually reproducing organisms, and the purpose is to reduce the chromosome number so that the offspring have the same number as the parents. Overview of meiosis. You're going to begin with one diploid cell, which, remember, for humans is 23 pair or 46 singles. It's going to go through 2 cell divisions instead of 1. At the end, you're going to get 4 haploid daughter cells, and haploid means half the number, so only 23 chromosomes in each daughter cell. The pairs of chromosomes in meiosis are called homologous or homologs. Meiosis 1. Homologs line up side by side at the equator and go through a process called synapsis. And when pairs separate, each daughter cell receives one member of the pair. And we'll talk more about synapsis as we go through the specific stages. The cells at this point are haploid cells. And then in meiosis 2, DNA doesn't go through interphase again, because we don't need to replicate anything. So the centromeres divide, the sister chromatids migrate to opposite poles, and they become individual chromosomes. And each of the 4 daughter cells produced have the haploid number of chromosomes. So here's a picture of, looks very similar to what it would look like in mitosis. These are called homologous chromosomes. So this is a pair of homologous chromosomes, here. They're going to get together and they're going to go through a process called synapsis. And they're going to wind around each other and actually exchange genetic information. The homologs go through synapsis, then they separate, and then the sister chromatids separate further into individual chromosomes. And then you have 4 haploid daughter cells. All right, fertilization. Daughter cells of meiosis become gametes. So sex cells are referred to as proper names as gametes. Sperm and egg are gametes. Fertilization restores the diploid number. So you take a sperm that has 23 and an egg that has 23, and you get 46 chromosomes for the new individual. Meiosis helps ensure genetic variation, and genetic recombination happens in 2 ways during meiosis. Goes through the process of crossing over during synapsis. Synapsis only happens in prophase 1. You need to remember that. And independent assortment, which is one of Mendel's laws of heredity, and we'll talk more about that in the next chapter. Synapsis of homologs. These are sister chromatids together here. This is a homologous pair. Crossing over occurs, and crossing over is exactly what it sounds like. They're going to actually exchange genetic information when they do that. And this just leads to more genetic variation for the sperm and egg that are made. So these are called recombinant daughter chromosomes because they don't look like the parents. They have the same thing. So I don't want you to think that right here codes for eye color and right here codes for hair color. And those are just silly examples, but that's not what it is. If this codes for eye color here, this section that was exchanged also codes for eye color. Independent assortment during metaphase 1. Look at the differences, the way these are lined up. When you look at these and they go through and they line up on the metaphasial plate, you can see these are the homologs got separated, but these are blue and blue, red and red, or it could be blue and red, red and blue. It doesn't make any difference. There's no set way that these are going to line up. In mitosis, they lined up in a line and the sister chromatids pulled apart, so you had the same thing on both sides, but this allows for genetic recombination when you see them independently assort during meiosis. All right, phase 1 of meiosis is prophase 1. This is where synapsis occurs. The nuclear membrane breaks down. The homologs line up side by side, and crossing over occurs during this phase. In metaphase 1, the homologs line up at the plate, and they tend to be in pairs so that it looks similar to metaphase in mitosis, but they're lined up in pairs instead of in singles. So you'll see here where we talked about mitosis, these would be in a straight line. They're actually lined up in their homologous pairs. Anaphase 1, the homologs, still consisting of 2 chromatids, undergo independent assortment into the daughter cells. And in telophase 1, it can happen, it may not happen, it just depends on the type of cell that's being reproduced and how quickly it needs to be reproduced. The nuclear envelope may reform, it may not. Cytokinesis may happen and have 2 daughter cells which are haploid. There's a period called interkinesis, and interkinesis is between meiosis 1 and meiosis 2, and there's no replication of DNA, so not interphase, but interkinesis, and it's kind of a resting phase. Meiosis 2, you have prophase 2, so the cells have one pair of chromosomes from each homologous pair left. Metaphase 2, the chromosomes line up in the metaphyseal plate. Anaphase 2, the centromeres divide and daughter chromosomes go toward the poles. And in telophase 2, nuclei reform, cytokinesis occurs, and you have 4 haploid cells. All right, the haploid cells are not identical to the parent cells. Combining of chromosomes from genetically different gametes, fertilization ensures the offspring don't look identical to parents at all. The genetic variability is the main advantage of sexual reproduction. This is why you don't look exactly like either parent, but kind of a blending of both. And in some, there's totally new facial features and things that neither parent has. DNA replication occurs only once prior to meiosis and mitosis. Meiosis requires 2 divisions, mitosis 1. Meiosis produces 4 daughter cells, mitosis 2. 4 daughter cells from meiosis are haploid, the 2 from mitosis are diploid. The daughter cells from meiosis are genetically variable, and those from mitosis are genetically identical. Meiosis only occurs at certain times of the life cycle, and that's to produce sperm and egg. Mitosis happens almost continually. It's really important that you be able to compare and contrast the stages of mitosis and meiosis, especially for the EOC. And that shows a good side-by-side picture, and this is in your textbook, that shows how meiosis forms 4 haploid cells and goes through 2 divisions, while mitosis goes through 1 division and has 2 diploid cells at the end. There's also a chart in your book that compares prophase 1 to prophase, metaphase 1 to metaphase, and so forth. Does the same thing in prophase 2. And remember, mitosis doesn't go through the second set of division, but there's still some similarities in prophase 1 and 2 with the phases of mitosis.